Whether you're a bioprocess engineer hunting for the perfect scaled-down model, a biotech founder architecting the leap to commercialization, or a scientific analyst who craves raw technical truth, you've reached the technical edge. In this space, we live by a single mantra: the process is the product. Welcome to the Biomanufacturing and Fermentation Technology podcast with Prasad Ernala. Hit subscribe and ring the bell to join a global community and stay at the vanguard of biotech translation. Let's build the bioindustrial future together.
This is the part 1 of our 4-part deep dive on design of experiments, where we explore how AI is reshaping DoE without changing the biological realities that make rigorous experimentation essential in bioprocess development. Lesson 1. Thinking in experiments. How AI changes DoE fundamentals. Biology is interactive, nonlinear, and noisy. OFAT fails because it ignores interactions like glucose times DO times pH that fundamentally reshape metabolic behavior. Classical DoE still wins. Fractional factorials and response surface methods remain the most defensible way to map CPPs to CQAs and define true design space. The real bottleneck is experiment definition. Objectives, factor selection, ranges, and meaningful responses matter more than statistical computation. Replication strategy beats raw sample size. Center point and temporal replication are critical in living systems where biological variability dominates. AI is a decision support tool, not an experimental designer. It accelerates design generation and analysis, but humans must own biological plausibility, regulatory alignment, and final process decisions.
Welcome back to the deep dive. Today we're wading into the messy, chaotic, and often just incredibly frustrating world of biological experimentation.
That is the perfect way to describe it.
We've got this whole stack of course materials here on something called design of experiments, or DoE. But before you tune out because you heard the words statistics, just stick with me.
Please do.
Because we are not just talking about math, we're talking about a collision.
It really is a collision. You have this, rigid, classical scientific method on one side, this stuff we've all used for centuries. And on the other, you have the absolute chaos of biology.
Right.
And now, thrown right into that mix, you have the speed of artificial intelligence. It's a volatile combination.
And looking at this first lesson from our source material, Thinking in Experiments, the main tension is, well, it's pretty clear. It's about why the way we were all taught to do science in high school is fundamentally broken.
Broken is a strong word, but yeah, in the context of modern biotech, it's accurate.
Yeah.
If you're trying to optimize, say, a fermentation process with the tools you learned in 10th grade chemistry, you're not just moving slowly. You're probably walking in the wrong direction.
And that's the one factor at a time method, OFAT. Now, I have to play devil's advocate here right off the bat.
Go for it.
Because the source material is really aggressive against OFAT. But intuitively, it feels safe. It feels responsible. I change the temperature, I see what happens. I change the pH, I see what happens. It's logical.
Right.
Why is the source calling this a trap?
Because it is logical, but only for simple linear systems. If you're fixing a car and you want to know which spark plug is bad, you check them one by one. That works. But biology is not a car engine. The source makes a really critical distinction here. Biology is defined by interactions and nonlinearity.
Nonlinearity. That's one of those words that gets thrown around in meetings to sound smart. But what does it actually look like when I'm staring at a bioreactor?
Think about consequences.
Yeah.
In a linear system, you double the input. You double the output.
Yeah.
Simple.
Right.
In biology, if you double the glucose feed to a microbe, you don't just get double the growth. You might trigger a stress response. You might cause the cell to switch its whole metabolic pathway. You might just kill it.
So the input doesn't just change the volume of the result, it changes the nature of the result.
Exactly. And that is where OFAT just completely blinds you.
Yeah.
The notes use a specific term, the keyhole view.
Okay.
If you only look at one factor, you're looking through a keyhole. You might see a chair in the room, but you miss the fact that the room is on fire.
Okay, that's a vivid image. Let's get specific, though. The source uses this really complex example with microbial metabolism to prove the point. It's the glucose by dissolved oxygen by pH scenario.
Oh, yeah. This is the classic interaction trap. It's the horror story of fermentation science.
Walk us through this, because if I'm a scientist, my first instinct is glucose is food. More food equals more growth. So I crank up the glucose.
Right. So say you run an experiment where you just crank up the glucose. You keep your oxygen constant, you keep your pH constant. That's what OFAT tells you to do. And you see the growth rate go up. Success, right?
I mean, ideally, I write down high glucose is good in my notebook and move on.
But then you try to scale it up. You add that high glucose. But because there's more food, the cells metabolize way faster. They chew up all the oxygen in the tank at a rate you did not anticipate. And suddenly, your dissolved oxygen, your DO crashes.
And now the cells are suffocating.
Right, so they panic. They switch to anaerobic metabolism just to survive. They start fermenting, maybe producing lactate or acetate. Now your pH starts to drop because of all that acid.
And most enzymes absolutely hate acid.
Exactly. So the very enzymes that process the glucose stop working because the environment is too acidic. So you added more food to help them grow, but because you didn't adjust the oxygen or the pH control at the same time, you actually killed the entire process.
I killed them by feeding them. And if I had tested those things separately, OFAT style, I would never have seen that coming. I'd have a note that says high glucose is good and another that says standard oxygen is fine.
And that is the interaction. The effect of the glucose depends on the oxygen level, which depends on the pH control. That's the dance. And you cannot find interactions by testing one thing at a time. It is physically impossible.
It seems so obvious when you lay it out like that, but in the heat of the lab, when you have a deadline, it's just so tempting to tweak one knob. It feels controllable.
It feels like control, but it's an illusion. DoE design of experiments is actually about gaining real statistical control over that chaos.
But let's be real about why people don't do it. The source talks about statistics anxiety, and I feel that. I hear DoE, and I immediately think of JMP software and multivariate analysis. Sounds like you need a PhD in math just to pipette some liquid.
It is a barrier,
Yep.
or it used to be. But the source material argues that the math isn't the real bottleneck anymore.
How?
We have software for that. We have AI for that.
Yeah.
The new bottleneck, the real struggle, is what they call experiment definition.
Experiment definition. That sounds kind of vague. What does that actually mean?
It means knowing what to ask. The software can crunch the numbers all day, but it can't tell you if you're asking the right questions in the first place. It all comes down to your factors.
Right, the inputs. But the problem isn't knowing what a factor is. The problem is I have 50 of them. Temperature, stir speed, 3 different media components. I can't test 50 factors in a DoE matrix. I'd be running experiments until 2050.
Exactly. And that's the screening problem. And this is where human expertise is so vital. You have to be able to differentiate between your CPPs and your CQAs.
Okay, let's elevate the jargon a bit here. CPPs and CQAs, these get thrown around in biomanufacturing constantly.
Right. So CQA is the critical quality attribute. That's your destination. I want a high yield of this specific protein, and it needs to be pure.
And the CPP.
Critical process parameter. These are the knobs, the levers, that actually move the needle on that quality attribute. The hardest part of DoE isn't the math. It's using your biological intuition to look at those 50 possible factors and say, okay, these 45 are probably noise. These 5, these are the CPPs. These are the ones we test.
So you're saying we need to stop worrying about the p-values so much and start worrying more about the biological mechanism?
Yes. You can run a statistically perfect experiment on the wrong variables and get a perfectly valid answer that is completely useless to your project.
Which brings us to the other nightmare of biology that the source focuses on, variability. The noise.
The noise is the enemy.
Yeah, the text distinguishes between biological variability and technical variability. I feel like scientists often just conflate these two, or just call it experimental error and wave it away.
We do, and it's dangerous. Technical variability is us. It's the machine calibration, the slight tremor in your hand when you pipette. The fact that the shaker table wobbles a bit more on the left. That's error.
And biological variability.
That's just life. That's the fact that 2 genetically identical cells in the exact same environment will sometimes just behave differently. It's not an error, it's the system functioning as designed.
So how does DoE handle that? Because if the system itself is noisy, how do you find the signal? The standard answer you always hear is, just increase N. Run 1 million samples.
N equals 1 million. The brute force approach.
Oh, that's so expensive. And if I'm pipetting by hand, my hand is going to fall off.
Not only is it expensive, but it can be really misleading. The source drops a very tactical point here. Replicate strategy matters more than sample size.
Explain that, because if I run 50 samples, that has to be better than running 3, right?
But not if you run all 50 on the same day with the same batch of media prepped by the same person.
Oh, because then I've just measured how that one person, that one media, and that one afternoon perform.
Exactly. You have effectively run N equals 1 50 times. You have zero data on how the biology performs on a different day or with a different batch. You have high precision. Your dots are all clustered together, but you have very low accuracy.
That is a painful realization.
The source suggests that 3 replicates spread across 3 different days is vastly more powerful than 10 replicates done all at once.
So you have to force the system to show you its true noise.
You have to bake the variability into the design. Only then can you actually trust the signal.
This feels like the perfect segue to the AI element, because there's a huge misconception, I see it on LinkedIn every single day, that AI can just clean up the data, that you can feed it messy, noisy, poorly replicated data, and the neural net will just find the truth.
That is the AI blind spot. And honestly, it is terrifying for the future of science if we don't get a handle on it.
Why terrifying?
Because an AI is a pattern recognition machine. It is designed, fundamentally, to find a pattern. If you feed it noise, it will find a pattern in the noise. It will hallucinate a relationship that doesn't actually exist.
So it won't say insufficient data. It'll say, hey, it looks like temperature correlates with yield, when really it just correlated with the fact that the AC unit broke that afternoon.
Exactly. AI cannot average out bad experimental design. If you stop thinking like a biologist and just trust the algorithm, you will chase ghosts. You'll spend 6 months optimizing a phantom signal.
So let's talk about the solution, the hybrid workflow. We're not Luddites here. We want to use these tools. But the source outlines a very specific mental model for how to do it safely.
It breaks it down into 3 steps: the human definition, the AI design, and the human check.
Okay, the first one, human definition. This goes back to what we said earlier. The bottleneck is knowing what to ask.
Right. You, the human, have to define the goal and the realism. And AI doesn't know that you can't heat a protein past 60 degrees without denaturing it, unless you tell it.
Or that you only have enough budget for 20 runs, not 200.
Exactly. Constraints are human.
Then comes the AI's role. Propose designs and offer interpretations. This is where the heavy lifting happens.
This is the combinatorics. I mean, you have 5 factors at 3 levels. That's a lot of combinations. The AI is brilliant at building that matrix, the actual table of experiments that maximizes information with the fewest runs.
But then, and this seems to be the part everyone skips, it loops back. The human check.
Validating biological plausibility.
Yeah.
This is the safety valve. This is everything.
The source actually has a hands-on exercise for this, specifically about prompting. I want to try this out right now. Let's do a live role play.
Let's do it.
Okay, the prompt they suggest is explain factor interactions using glucose, temperature, and pH in yeast fermentation. Sounds like a standard homework question.
It does. Very standard.
So I typed that into my LLM, and let's say the AI spits back this. Higher glucose increases energy availability, leading to higher biomass. Higher temperature increases reaction rates, speeding up growth. Optimal pH ensures enzyme function.
Yeah, okay, stop there.
What's the verdict?
It's technically true, but it's biologically naive. It reads like a textbook from 1980.
So as the expert, if I hand you that as the basis for a design, what are you tearing apart?
I'm looking for the stress responses. Did the AI mention the Crabtree effect?
So?
The Crabtree effect.
Crabtree effect.
In yeast, if the glucose concentration is too high, even if there's plenty of oxygen, they'll switch to making ethanol alcohol, which is toxic to them. It actually limits biomass. The AI said more glucose equals more biomass. That's just wrong in the real world.
Okay, what else?
Temperature. The AI said higher temperature equals faster growth. But higher temperature also increases protease activity, the enzymes that chop up other proteins. If you're trying to make a sensitive protein product, high temperature might grow the cells fast, but they'll just chew up your product as soon as they make it.
So the AI gives you the happy path, but biology is rarely happy.
Exactly. If you just take the AI's design based on that simple understanding, you might design an experiment that kills your yeast or destroys your product. You need to look at the output and say, that's too simple. Add constraints for ethanol toxicity. Add constraints for protease activity.
You have to complicate the model.
You have to inject the biotech engineering reality into the biology 101 model that the AI defaults to.
That's a really helpful way to frame it. You are the realism constraint.
Yes.
There's a list here in the notes of what not to delegate. I think this is crucial. We can delegate the math, the matrix generation. What can we never, ever delegate?
Why? The scientific intent. You cannot ask an AI what is interesting about this molecule. You have to know why you were studying it.
And you can't delegate the sanity check.
Never. If the AI suggests a run condition that is physically impossible, like a flow rate your pump can't handle, or a concentration that would turn the media into syrup, you have to catch that. The AI doesn't know your pump is broken.
It's like having a brilliant intern who's read every book in the library but has never actually stepped foot in a wet lab.
That is the perfect analogy.
Yeah.
They know the theory, but they don't know that the shaker table creates shear stress that bursts the cells at 200 RPM. You have to provide that context.
So, looking at the outcome for this lesson, the goal is to move from design generation, just making tables, to decision support.
It completely changes your value as a scientist.
How so?
Well, 10 years ago, if you could do the math for a fractional factorial design by hand, you were a wizard. You were indispensable.
Right.
Now, a plugin does that in 3 seconds.
So that skill is devalued.
Completely. Your value now rests entirely on your ability to spot biological naivety. Your value isn't asking the question that exposes the gap in the model.
It sounds like we're moving from being calculators to being critics.
That's it. And frankly, that is a much harder job. It requires deeper biological knowledge. You can't hide behind the math anymore. If the experiment fails, you can't blame the statistics. You have to blame your understanding of the biology.
That's daunting, but also kind of liberating. It puts the science back in the driver's seat.
It does. It forces us to be better biologists.
So, for the listener who's holding a pipette right now, wondering if they should try DoE or just stick to changing one thing at a time because it's easier, what's the takeaway?
The takeaway is that complexity is unavoidable. Biology is complex. You can ignore that complexity with OFAD and fail slowly, spend months troubleshooting a problem you don't even understand, or you can embrace it with DoE and AI and learn quickly.
Fail slowly or learn quickly. I like that.
The AI is a lever. It multiplies your force, but you have to know where to place the fulcrum. And the fulcrum is your biological intuition. If you place it wrong, the lever doesn't work.
And if you lean on it too hard without checking the base, it snaps.
Exactly.
We're going to wrap it up there. This has been a dive into the hybrid workflow. It's not about AI replacing scientists. It's about AI forcing scientists to actually be scientists again.
Couldn't have said it better myself.
One final thought to chew on before we go. We talked a lot about us checking the AI. But if AI is generating the hypotheses and the designs, and you are just the constraint checker, at what point does the AI start checking you? When does the model know more about the biological noise in your system than you do?
That is the scary question for next year.
Something to think about. Thanks for diving in with us. We'll see you next time.
Hi there, thank you for staying with me until the very end. If these technical insights help bridge the gap in your own process, please hit subscribe and make sure to hit the notification bell. Stay connected for our next deep dive into the realities of biomanufacturing. I am Prasad Ernala. Thanks for listening.